Our bodies are like bustling cities. Cells are the citizens of these cities. They communicate with each other constantly. This communication is vital. It keeps our bodies functioning properly. Cells use chemical messengers to talk to each other. These messengers can be hormones, neurotransmitters, or even gases. These messengers don't always enter the cell directly. Instead, they often trigger a cascade of events. This happens inside the cell. These events involve special molecules called secondary messengers. Secondary messengers amplify the original signal. They also relay it to different parts of the cell. Think of it like this. The original messenger is like a knock on the door. The secondary messenger is like a chain reaction inside the house. This reaction turns on lights, opens windows, and prepares a meal. Secondary messengers are essential for life. They regulate a wide range of cellular processes. These processes include metabolism, growth, movement, and even death. Understanding secondary messengers is key to understanding how cells work together. This knowledge is crucial for developing new drugs and therapies. Cells have specialized proteins on their surface called receptors. These receptors act like antennas. They pick up specific signals from the outside world. Each receptor is designed to bind to a particular messenger molecule. This binding triggers a change in the receptor's shape. This shape change is the first step in signal transduction. Signal transduction is the process of converting an external signal into a cellular response. The activated receptor then interacts with other proteins inside the cell. This interaction sets off a chain reaction. Think of it like a lock and key. The messenger molecule is the key. The receptor is the lock. Only the right key can unlock the receptor and initiate a response. One of the most important secondary messengers is cyclic AMP, or cAMP. cAMP is produced when a hormone like adrenaline binds to a G-protein-coupled receptor, or GPCR. GPCRs are a large family of receptors found on the surface of many cell types. The binding of adrenaline to its GPCR activates an enzyme called adenylate cyclase. Adenylate cyclase converts ATP, the cell's energy currency, into cAMP. cAMP then acts as a second messenger. It activates other enzymes called protein kinases. Protein kinases are like molecular switches. They turn other proteins on or off by adding a phosphate group to them. This process, called phosphorylation, can dramatically alter a protein's activity. A classic example of cAMP in action is the fight-or-flight response. When we encounter a threat, our adrenal glands release adrenaline. Adrenaline binds to GPCRs on liver cells. This binding triggers the production of cAMP. cAMP then activates a cascade of protein kinases. These kinases ultimately lead to the breakdown of glycogen into glucose. Glucose is the primary source of energy for our cells. This rapid release of glucose provides our muscles with the fuel they need to fight or flee. cAMP also plays a crucial role in regulating heart rate, blood pressure, and other physiological responses during the fight-or-flight response. Section 5. cGMP, the smooth operator of blood vessels. Another important secondary messenger is cyclic GMP, or cGMP. It's quite similar to cAMP in structure and function. cGMP is produced by an enzyme called guanylyl cyclase. Now, guanylyl cyclase is activated by various signals, including nitric oxide, or NO. cGMP plays a crucial role in relaxing smooth muscle cells. Smooth muscle is found in the walls of blood vessels, the digestive tract, and other organs. When cGMP levels rise, it activates an enzyme called protein kinase G, or PKG. PKG then phosphorylates proteins involved in muscle contraction. This phosphorylation leads to muscle relaxation, and this is how drugs like nitroglycerin work to relieve angina. They increase cGMP levels, which in turn relaxes blood vessels and improves blood flow. Section 6. Calcium ions, the rhythmic drumbeat of cells. Calcium ions, or Ca2+, are not technically secondary messengers. They are ions, not molecules. However, they play a crucial role in cellular signaling. Cells maintain a low concentration of calcium ions in their cytoplasm. This is achieved by pumping calcium ions into storage organelles like the endoplasmic reticulum, or ER. When a signal arrives at the cell surface, it can trigger the release of calcium ions from the ER into the cytoplasm. This rapid increase in calcium ion concentration acts as a signal. It activates a wide range of calcium-binding proteins. These proteins then go on to regulate various cellular processes. These processes include muscle contraction, neurotransmitter release, and gene expression. Section 7. IP3. The Keymaster of calcium release. One way that cells release calcium ions from the endoplasmic reticulum is through a molecule called inositol trisphosphate, or IP3. IP3 is produced when a signal molecule binds to a receptor. This receptor activates an enzyme called phospholipase C, or PLC. PLC then cleaves a membrane lipid called IP2 into IP3 and diacylglycerol, or DAG. IP3 then diffuses through the cytoplasm and binds to IP3 receptors on the ER membrane. These receptors are calcium channels. When IP3 binds to them, they open, allowing calcium ions to flow out of the ER and into the cytoplasm. Section 8. Nitric oxide, a gaseous messenger with powerful effects. Nitric oxide, or NO, is a unique secondary messenger. It is a gas, which allows it to diffuse freely across cell membranes. NO is produced by the enzyme nitric oxide synthase, or NOS. NOS converts the amino acid arginine into NO. NO activates guanylyl cyclase, the enzyme that produces cGMP. This activation leads to increased cGMP levels and subsequent smooth muscle relaxation. This is how NO regulates blood pressure, blood flow, and other physiological processes. NO also plays a role in neurotransmission, immune response, and cell death. Section 9. Diacylglycerol, the orchestrator of immune responses. Diacylglycerol, or DAG, is another important secondary messenger. It is produced alongside IP3 when PLC cleaves PIP2. DAG remains in the cell membrane. It activates an enzyme called protein kinase C, or PKC. PKC is involved in a wide range of cellular processes. These processes include cell growth, differentiation, and apoptosis. DAG plays a particularly important role in regulating immune responses. It activates immune cells like T cells and B cells. It also regulates the production of cytokines, which are signaling molecules involved in inflammation. Section 10. Maintaining cellular harmony, the ensemble of secondary messengers. Secondary messengers are essential for life. They allow cells to respond to their environment in a coordinated and efficient manner. By amplifying and relaying signals, secondary messengers ensure that cells can communicate effectively. This communication is crucial for maintaining cellular harmony. From the fight-or-flight response to blood vessel relaxation, from muscle contraction to immune responses, secondary messengers orchestrate a symphony of cellular events that keep our bodies functioning properly. Understanding the intricate workings of these molecular messengers is essential for unraveling the complexities of human health and disease. It also paves the way for developing new drugs and therapies that target these signaling pathways. As we delve deeper into the world of secondary messengers, we gain a greater appreciation for the elegance and efficiency of cellular communication.